I welcome you to organic chemistry class. Today we are going to take biomolecules, that is chemistry in biology and medicine. So in medicine we have so many organic chemistry we are using. Some of the examples and the categories we will take. Similarly, the molecules which are involved in the biological reactions, the prominent molecules also we will take. Under this biomolecules we have carbohydrates first. Examples like glucose. Glucose is a carbohydrate which has a formula C6H12O6. In fact, this also can be written as a 6CnH2ON. That is where N is the 6 if you take 6 carbons and H2O6. Okay, that means it looks like 6 water molecules are attached to the carbon. That's why they named as a carbohydrates. But later on it is proved it is not just the 6 water molecules, but glucose has a structure of an aldehyde. Glucose is primarily an aldehyde containing 6 carbons. On each carbon we have one OH. So more than one OH, the compounds will become unstable. So we have one OH on each carbon, rest are the hydrogens. This is the structural formula of the glucose. So we have an isomer for the glucose. At the second carbon, if a carbonyl group is there, first carbon CH2OH, the last carbon is also CH2OH. The middle carbons OH and OH is common. This is called fructose. So if you see carefully, the glucose is an aldehyde, fructose is a ketone. This is a aldehyde. They are defined as the carbohydrates are nothing but the carbonyl compounds containing polyhydroxy, and there is an optical activity. There is a 5 carbons here are showing the optical activity. 5 carbons are optically, no, 4 carbons are optically active. In this 2 carbons are optically active. Okay, 3 carbons are optically active. Here 4 carbons are optically active. So optically active polyhydroxy carbonyl compounds are known as carbohydrates. Among them, this glucose and fructose are the most important molecules. Now the carbohydrates can be classified. Classification of the carbohydrates. Carbohydrates are divided into the 2 types. The old classification is sugars and starches. This is one method of classification. Sugars are soluble in the water and the starches are not soluble in the water. Later, the carbohydrates are divided into in a different way. Carbohydrates are divided into the mainly 3 types. Monosaccharides. Saccharide refers to a carbohydrate unit. Monosaccharide, oligosaccharide and polysaccharide. Monosaccharides are glucose and fructose. Oligosaccharides are di, tri, tetra, up to 10 you can take it as under this maltose, cellulose, raffinose, and polysaccharides, starch, cellobiose. Cellobiose is the disaccharide, not cellulose, cellobiose. Cellulose is a polysaccharide. Cellulose is a polysaccharide. So this is based on the classification. When you hydrolyze this disaccharide like cellobiose or maltose, when you hydrolyze, you will get the 2 glucose units. So that's why they are called disaccharides. If it is giving a 3 monosaccharides, trisaccharides. So up to 2 to 10 we have defined as oligosaccharides. In some books, monosaccharide, disaccharides and oligosaccharides also we have. Poly means more than 10 monosaccharides if it are there, this is called polysaccharide. Okay. The monosaccharides are on hydrolysis, there is no further the breakage, such a single piece type of the compounds, carbohydrates are called monosaccharides. The monosaccharides also can be classified into the 3-4 varieties based on the number of carbons present in that compound. The smallest carbohydrate is 3 carbon, one is known as triose, and 4 carbon one is called tetrose, pentose and hexose. This that means a triose means 3 carbons containing one, tetrose means 4 carbon containing, 5 carbon containing. In the hexose we will get this glucose and the fructose. In pentose we have ribose, which is a important one in the DNA RNA construction. In tetrose we have erythrose and threose and so on. Triose glyceraldehyde is the example. Even this the hexoses also we have further classification. If you take hexoses also further divided into the aldoses and ketoses. Aldoses, glucose is example. Ketoses, fructose is the example. So glucose is an aldose. Glucose is an aldose because it has an aldehyde function. This is based on the carbonyl function nature we have classified. Glucose is an aldose. Glucose is an hexose because it is made up of 6 carbon one. Glucose is a monosaccharide. On hydrolysis it won't break up. Glucose is a carbohydrate. Okay, it is a carbohydrate containing monosaccharide nature, containing hexose nature, containing it is a aldose. It is an aldehyde function containing one. So the glucose, fructose are important for the biological, that is in the biology of the carbohydrates. Carbohydrates are the ready energy for the in the human body or the in biological systems. Carbohydrates oxidize to the ethyl alcohol, afterwards it is oxidized to the carbon dioxide, it gives a energy for the body. So therefore carbohydrates makes an important role in the this one. Then we have a proteins are the second type of the molecules. Proteins also, like carbohydrates, the polysaccharides are the polymers. Just like that, proteins is also a polymer. This proteins contains amino acids made up of with the amino acids are the units in the proteins. Amino acids are classified into the 2 types, natural amino acids and synthetic amino acids. Synthetic means man-made, natural means either in the plants or animals we get. Natural amino acids are again divided into the 2 types, alpha amino acids and rest type of the other type of the amino acids. It is interesting to observe all the natural happens to be alpha amino acids. What is alpha amino acid? A carboxylic acid, when you have this is the functional group, leave the functional group, the second carbon is called alpha carbon, third carbon is called beta carbon and fourth carbon is called gamma carbon and so on, it is numbered in the alphabet. So therefore the amino group is on the second carbon. Amino and the carboxylic are both are attached to the same carbon. Such a type of amino acids are alpha amino acids. All the so far identified 24 amino acids are identified as a alpha amino acids only. Amino acids also classified on the basis of the basicity, basic and acidic nature also. They are divided into the neutral amino acids, Neutral means it will contain one carboxylic acid, one amino group. So therefore, the acidity and the basicity are compensated. Such amino acids are neutral. Whereas the acidic amino acids are, the amino acids will have a carboxylic as well as amino. In addition to that, there will be a branch, that branch also contains one extra carboxylic acid. Therefore, the amino acid exhibiting the acidic nature. Similarly, basic amino acids. In the basic amino acids, we will have a acid and amino, and this the group will have after some n carbons we have a amino group. So this amino and this amino, two amino groups are there, for it shows the basic character. So amino acids can be three types. Amino acids also can be classified based on the, will it contain a aromatic or not, non-aromatic and the aromatic, open chain, cyclic. In the cyclic also heterocyclic, homocyclic and so on, sulfur containing and non-sulfur containing. Amino acids also classified as a essential amino acids, and non-essential amino acids. Essential amino acids are around 8 to 10 amino acids we are mentioning under this category, and these essential amino acids are essential for the body, and these essential amino acids body cannot synthesize. Therefore, we have to take these amino acids from the outside in the form of diet. Whereas non-essential amino acids, they are also essential for the body, but body can synthesize from the essential amino acids or from some other compound. They can synthesize. The body can, body means here human body only reference, human body can synthesize these amino acids. Such amino acids are called non-essential amino acids. Now these amino acids are going to link up and there comes this protein molecule. Let us take some examples like a CH with a CH3 with a COOH. Okay, this is called a alanine. Alanine is a alpha amino acid. It is a natural amino acid. The carbon has an optical activity, will have a LEO, the optical, the rotation also. This amino acid is linked with the another that is a amino acid. Then what happened? The OH of this one, hydrogen of this one is removed as a water. So two amino acids are reacting, one acid group of the first amino acid and amino group of the second amino acid is going to react, and there comes one CONH grouping. CONH grouping. This type of CONH group is known as a amide bond. Chemically it is known as amide bond. This is known as amide bond. But biologically it is known as peptide bond. It is known as peptide bond. Now how many amino acids are reacting? 2. 2 gives one unit. This is called dipeptide. So how many amino acids are there? 2 are there. That's why it is called dipeptide. In dipeptide, how many peptide bonds are there? Only 1 is there. If A, B, C are linked, 3 amino acids are linked, then how many peptide bonds will be there? Here 1 peptide bond, here 1 peptide bond. So a tripeptide will contain a 2 peptide bond. So peptide bond is nothing but the NHCO. This can be hydrolyzed with dilute acid or enzymatically to give back the original, the amino acids can be prepared. Like that, one more amino acid if it is linked, then you will get a tripeptide and so on, you will get a polypeptide. Similar case, if it is repeated, several amino acids after joining, you will get the polypeptide. In this polypeptide, there will be a sequence, isn't it? 1, A, B, C, D and so on, isn't it? So if the sequence you have identified, what is the order of the different amino acids joined? If that secret, if you know it, that structure is called a primary structure. Primary structure. Primary structure. So a polypeptide, the order is called a primary structure. This primary structure, after getting, it may turn up slowly into a protein. A protein will come not just by the attachment of the ABCD, but this ABCD, the chain may go on coil up like this. Okay, then between this chain to here, there may be some hydrogen bondings like NH and CO is there, isn't it? The CO minus and H plus will have a hydrogen bonding. So due to the hydrogen bondings, the line may undergo a coiling. This coiling formation is called secondary structure. It's called secondary structure. So secondary structure is deriving through the intermolecular, within the molecule there comes some hydrogen bondings and secondary structure is formed. Now the secondary structure, if it looks like a rope type, so sometimes you have a rope, if you cross sect the rope, there will be a 6 or 7 small ropes you may find. These small ropes are like this, they are twisted in fact. You may find outside a rope like this big. When you cut this rope, you find there 6 or 7, sometimes middle hollow will be there. This type of the ropes, 6 or 7 will twist up and will make a big structure. Now between a coil and a coil, there comes some hydrogen bondings and ionic bonds and so on. Internally the dipole attractions, all such attractions will be existing between this one, and they are also twisted each other. Therefore based on the central core, the remaining this is all actually single molecule. It goes up and down, up and down, up and down. It makes a big rope type of the structure. This twisted structure, that is rope type of structure, will give the tertiary structure. A tertiary structure. This tertiary structure again is responsible through the hydrogen bonding, and within the molecule, between the rope and the rope, there will be a different types of the attractions that makes a rope type of the structure. Imagine it is only just the visualization, but this rope type of structure, if you take a purchase, let us say 10 kgs of a rope, if you put it on a corner, it may take a specific shape containing some gaps internally and so on. This the rope will be having a internal structure and all that. So there comes some specific attractions between the rope and the other ends of this one, and that takes it to a fourth level of structure. It is called a quaternary structure. The quaternary structure will have a specific three-dimensional caves. Into that a specific molecule, let us say D-glyceraldehyde enters into that cave. L-glyceraldehyde will not be fit. So systematically the structure, three-dimensional structure is developed, that is called quaternary structure. So if all this is developed, then it is called protein. A protein is not a simply chain of the polypeptide. Polypeptide is a simple chain. If the polypeptide undergoes this primary, secondary, tertiary and quaternary structures, it becomes a protein. Some of the proteins are acting as hormones, right? Some of the proteins are body building materials, some are acting as vitamins and so on. Different the work, the proteins are catalysts, some are working as enzymes. Some are working as enzyme. Most of the enzymes are nothing but the proteins. All proteins may not be enzymes, but all the enzymes, biological enzymes, are nothing but the proteins, proteinous material. How come this enzymatic action means the quaternary structure is responsible? If you heat it, or put some more acidic or basic conditions, the internal hydrogen bondings will be break up. High temperature beyond the 50 degrees centigrade, people have vomiting and all that. This is because of the denaturation of the protein. denaturation of the protein structure, not the primary structure, especially the quaternary, tertiary and secondary structure is lost, then biologically it will become not fit to do this all these enzymatic actions. So therefore the whole biological catalysis, the metabolism will be upset down, and then person will get a vomiting and the motions and all that will be taking, that is dehydration taking place because of this denaturation of the protein. So the protein is not just a polymer of amino acid, but this all this 4 level structure is also important for this protein. Then we have another important biomolecule is the nucleic acids. Nucleic acids. These are also, all these 3 are polymers actually. Carbohydrates, especially in terms of the starch and cellulose, is a polymer, biopolymers these are all. Okay. So nucleic acids are made up of with one sugar unit will be there, and it is linked with one organic base will be there, and this sugar is linked to a phosphate. This will be n times will be there along the chain of the sugar and a phosphate and a base. Okay. Sugar, what type of sugar will be there? It will be generally a ribose will be there. That is a 5-membered ring will be there, a cyclic form of the 5-membered ring will be there, and here a 1 carbon, that is 4 carbons, and the oxygen will be 5-membered ring, and 1 carbon will be out here, that is the first carbon and the 4th carbon is internally linked, a hemiacetal structure is there. This type of ribose will be there. Okay. At second carbon we have a OH, so it is a ribose. If this OH is missing, it is called deoxyribose. Deoxyribose. Both ribose and deoxyribose are there as a sugar unit. What is phosphate? Phosphate is nothing but a PO4 unit, a PO4 unit. This is linked to the sugar, that is linked to the another sugar. Continuously another sugar will be there, that will be having a base. What is this base? The bases are 2 types are there. Bases are 2 types. The bases can be containing a 6-membered ring with 2 nitrogens like this. Okay. This is called pyrimidine type of the bases. Pyrimidine type of base. Another type is there is a 6-membered ring with pyrimidine and also connected with a 5-membered ring containing 2 nitrogens. This is also aromatic system will be there. So this is called purine type of the. So there are 2 purines and the 3 pyrimidines are there and 2 purines are there. Okay. So this is now the one of the base will be there. We can't say which base will be there, but one base, either a pyrimidine or a purine will be there, and this will make a long chain. That long chain is called what? A nucleic acid. These nucleic acids, that's why when you hydrolyze with either with the basic hydrolysis, that is acidic hydrolysis, or enzymatic hydrolysis, you will get smaller units like this one. That is a base, sugar and phosphate containing unit may be generated. Sugar and phosphate and base containing units, these are called nucleotides. The nucleotides further the hydrolysis will give only the sugar and the base. And phosphate will be out. Now this is a connected one. Okay. Sugar, phosphate and base connecting units are called nucleotides. When hydrolyze, phosphate will be out and sugar and base units will come. This is called nucleosides. Nucleosides, when you hydrolyze further, you will get a sugar and further a base will be there. So you can identify which base and which sugar. So it is go on degradation, a polymer. This is called polymer. Sugar, phosphate, base is a polymer. What is the name of this polymer means? The polymers are 2 types. One is RNA and the other one is DNA. What is the difference between RNA and DNA? DNA present in the nucleus. This is the First difference. This is present in the nucleus plus in biological outside the nucleus also may be there. In the nucleus as well as outside the nucleus also RNA will be there. So, DNA will be only in the nucleus. By occurrence we are having one difference. In the DNA we have a deoxyribose. This is the second difference. This will have a deoxyribose. Deoxyribose. Here ribose, in the RNA you have a ribose. This is the second difference. A ribose is a 5 carbon unit containing the aldehyde function. The last one is CH2OH, second OH, third OH, and this one. At the second OH, we have this OH function. This OH, if it is there, it is called ribose. Instead of OH, if you have a hydrogen without oxygen, that is called deoxyribose, which you find in the DNA. Right. In addition to this one, the purines and pyrimidines will be there. Will be this will be 2 lines of nucleic acids will be in the form of a double helix manner. Double helix manner. Watson and Crick identified the structure of the nucleic acid. The nucleic acids will be double helix manner. They will be twisted, and in between this one, a guanine G and combines with C, guanine G, C, T versus A. Okay. So, in the RNA, this is in the DNA you find this type of the sets you find in the DNA. In the RNA, what happens, instead of thymine you will find a uracil. So, therefore, another difference is in DNA you will have a thymine. In the RNA you find a uracil. Okay. And these two are adenine, these are purines. These 3 are the pyrimidines. So, there are 3 pyrimidines I said. One is cytosine, one is thymine, and the other one is uracil. Guanine and adenine are the purines. So, this out of the 5, this one, there will be a combination will be there, and this base and another chain with the base, at the base and base will have a, and G and C will have a 2 hydrogen bondings, and T and A will have a 3 hydrogen bondings. With this, the double helix is having a firmly attached to each other, and this double helix, whenever you want a new cell want to be created, a part of the nucleic acid will be removed, that is called replication, and that will transfer the information to the body to have a protein synthesis or to have a new cell, that is called transcription. A new information is transferred, that is called transcription. All these you find in the nucleic acids. It's a big molecule containing a small nucleotides, polynucleotide. Nucleic acid is nothing but a polynucleotide. Proteins are nothing but the polyamino acids. So, one nucleotide will have a sugar, phosphate and base. Bases are 2 types are there, pyrimidines and purines. There are 2 purines called the adenine and guanine, and 3 pyrimidines that is thymine and cytosine. In that RNA, thymine won't be there, instead uracil will be there. Okay. So, the combination always goes one chain, if guanine is there, that will be hydrogen bonded with the cytosine. In the other chain, if thymine is there, it will be attached to the purine, that is adenine only. So, like that the long chains will be attached in a double helix manner, that is called identified by the clock, that is the Watson and Crick. They won a Nobel Prize for this the introduction. So, the DNA will preserve a code system, that the codons will have a transferred into the information, and that is called actually genetic code. Genetic code means at what age what's going on, you need not to teach the biologically, that is inheritance, the coming to the systems, that is one person to the other person, how the information carried on means through the genetic code. Now, we have some molecules like vitamins, hormones and lipids. Small information let us take. Vitamins are small units, they are controlling specific activity, like we have vitamins A, B, a set of B is a complex actually, B1, B2 up to B12 we have, and vitamin C, D, E, K are the important vitamins. A is also known as retinol. A is also known as retinol. The deficiency leads to give the blindness. We have a complex of the B1, B6 and so on, thiamine, pyridoxine and so on. Vitamin B12 is called cyanocobalamin. Cyanocobalamin. Cyanocobalamin is a in the B complex, it is B12 cyano. Cobalt plus 3 will be there in this complex molecule it is, and it is controlling the blood preparation and so on. The C is known as ascorbic acid. Ascorbic acid. The deficiency leads to give the scurvy disease. Vitamin D is called calciferol, and deficiency leads to the rickets. Vitamin E is operating the reproduction, the malnutrition in the reproduction. Okay. Tocopherol it is called actually. Vitamin K is controlling the blood clotting. Hemophilia, resulting hemophilia, blood won't clot continuously, blood will be oozing. So what is the name of that vitamin, if at all it is there, like ascorbic acid, retinol, you try to remember, and what is the deficiency caused by these vitamins, also some of the questions coming. Hormones are also, these are the small the molecules to large size molecules are there. They are secreted by the ductless glands. Ductless glands, they are secreted by the ductless glands, like pancreas creates insulin and so on, and they are also biologically, they are very important. Hormones are simple protein molecules are there, like insulin. Some are the steroids. In the steroids we have a sex hormones, like female sex controlling and the male sex controlling, and the other one is the pregnancy controlling hormones. Female hormones are known as estrogens, male hormones are called androgens, and pregnancy controlling is and that is prednisolone and so on. Such a type of the steroids come under this category, isn't it? These are hormones. In the lipids we have lipids are actually oils and so on are the simple examples, but the lipids constitute a storage of the food, whereas the carbohydrates are ready-made energy. Lipids are there to store the energy, is the best this one, because the biologically they are very stable. So lipids will contain ester functions actually, but the lipids can be classified into 2-3 varieties. First variety will be simple category. In this simple category we have triglycerides and waxes. The large chain will be an alcohol group will be there. So many 20 to 30 carbons will be there and alcohol will be there. The triglyceride, there will be a glycerol will be there, and the glycerols are attached to an ester. A long chain alkyl group will be there. Okay. The alkyl chain will be so long, and when you hydrolyze 3 pieces of acids and 1 piece of glycerol will be containing, they are called triglycerides. And then we have a compound type of lipids. Compound type of lipids. In the compound type, we have a glycolipids and phospholipids. That means they may be attached to a carbohydrates, they may be attached to a phosphate. These are called phospholipids are adenine triphosphate, guanine triphosphate, like that. Phospholipids some also important. In the third type will be the derived one. Derived lipids. The derived lipids also 2 types, prostaglandins and steroids. Under the steroid, cholesterol is one important, and cholic acid is another important steroid. They are also having the biological activities, however, but mainly the lipids also work as a membranes. In the membranes, a membrane is constituted with proteinous layers and non-proteinous layers. In the lipids also can make one layer after the layer. Multi-layer membranes will be there in the skin. One of the layer will be the lipid layer. Okay. So, these are all biomolecules in fact. Now, coming to the medicinal importance containing biomolecules. Some are like analgesic. These are medicine important, the biomolecules which are continued, that is synthetically we made outside, but it is used into the biology. Analgesic drugs are divided into the 2 types, narcotic and non-narcotic. Under the narcotic, we have morphine, codeine, and heroin. These are the some narcotic drugs. Narcotic means when you take these drugs, analgesic means painkiller. Actually, it's a painkiller, but painkillers can be, the morphine also can, that is pain will be killer, but it will become the addictive slowly, and it is badly influencing central nervous system. Therefore, it is not advisable to go for, but unfortunately so many people will become addicted to this one. Non-narcotic will be the aspirin, ibuprofen are the good example for this. The analgesic. Analgesic means painkillers. They are painkillers. By definition, they are analgesic means painkiller. Antipyretic. Pyretic means, pyro means temperature, isn't it? Temperature removers are slow downing materials, isn't it? Antipyretic. So, analgin is one. Paracetamol and phenacetin. These are the important antipyretic, means temperature controlling the medicines are the drugs. Then antibiotic are the penicillin, and chloramphenicol and sulfadiazine. Penicillin, chloramphenicol, sulfadiazine, they attack on the microbes like bacteria and the virus, they kill them. So, therefore, they are working as a antibiotic. septic. Antiseptic are chloroxylenol, thionyl, bithionyl, and antiseptic and antihistamines, pheneramine, avil type of the drugs are antihistamine. Antihistamine means the rash, all such things, allergic produces. Septic means in the body, if there is a cut or so, the leads to give some attack of the fungal material or the biological system. In such a case, they kill that and this works as a antiseptic. Antihistamines and we have some more types like working as a some examples. Let us see in the different questions asked. In that we can go for the further examples and the discussion. Benzene 4-hydroxy acetanilide belongs to which of the family? This is nothing but a paracetamol. It is para amino, that is amino. Let us take here OH here. So, this is aminophenol. Aminophenol is a anti-pyretic drug. The amino group is acetylated, COCH3. This is called acetaminophenol. Acetaminophenol goes to the antipyretics. So, it is 4-hydroxy group, acetamino group, paracetamol. It is called commercially called paracetamol. If the hydrogen is replaced by a CH2CH3, it becomes a ether. Then it will become a phenacetin. It is called phenacetin. Is also one of the example. It was recently asked in the 2012 antipyretic. It is known as, right. Next. The site of action of insulin is, it is where it is working, isn't it? It is insulin is a that is a duct, this one. So, therefore, it goes into the bloodstream, it is into the plasma membrane, it is working. So, answer 3 is the right answer. It is a hormone. Hormones by definition are the ductless glands secretions there. So, therefore, it goes into the bloodstream and where it works in the membrane. Next. Which one of the following statements is not correct? Recently asked, this is also. So, except glycine, all other naturally occurring alpha amino acids are optically active. That is 100% true. It is. Alpha amino acids have maximum solubility in their at their isoelectric point. This is wrong. At isoelectric point, they exist as a plus minus Zwitter ion structure. These Zwitter ions are less soluble when compared with the only cationic and only anionic structures. A tripeptide has 2 peptide bonds. That is what I said, 3 amino acids joining gives a tripeptide. In between you have a 2 peptide bonds. Alpha amino acids exist as Zwitter ions at their pH, the isoelectric point. That is also correct, except the 2, all are right answers. Next. Which one of the following is an artificial sweetening agent? Artificial sweetening agents. Actually, we use sucrose that contains glucose and fructose, that is cane sugar. We crystallize out a sucrose molecule we are using, but artificially there are 3 materials, that is aspartame, allitane and sucralose are given in the syllabus. But you can also remember saccharin. Saccharin So, these all the 4 are artificial. This one, when a person is suffering from the sugar disease, that is a diabetic disease, this the other than the sucrose usage will be the better treatment. So, therefore, aspartame, alletane and sucralose and saccharide. This is the last one is called aspartame. It is a derivative of one of the amino acid called aspartic acid. So, the fourth one is the right answer. It is a long structure, but it is nothing but the aspartame it is. If the name is given, it would have become a right easy to identify. So, the answer 2, which of the following is an artificial sweetening? No, answer is given wrong. It is the fourth one is the right answer. Aspartame is the right answer. Next question. The ATGC ratio in human beings is, that is adenine thymine versus guanine cytosine is around 1.52. It may not be that important, but the question has come. It is 1.52 is the right answer, it seems. Next. Identify the non-narcotic analgesic from the following. I said narcotic are morphine, codeine, thebaine, and these are all narcotic drugs. Non-narcotic are that is aspirin is the non-narcotic. Ibuprofen and aspirin is given as the non-narcotic. From this, I think he has given one question. Ibuprofen is the non-narcotic analgesic, painkiller it is. Next. How many tripeptides can be prepared by linking the amino acids glycine, alanine and phenylalanine? Let us code this glycine as A, okay? Glycine as A, and second one as a B, and third one as a C. So, this A, B, C together is known as a tripeptide, right? So, you can also have a that is B in the center with A and C, isn't it? Similar the way B in the center, this side C, this side A is also different amino acid. This is a B with amino group, this side amino group, B with amino is attached to A, acid is attached to C is different, amino this side and acid this side if you take, these two are different, like that. B centered two peptides, tripeptides, A centered also, A with one side B, one side C. Now, you can have A with C and B. So, A centered 2, B centered 2, C centered also you can have A and B and B and A. Totally how many? So, 2 plus 2 plus 2, total 6 different the tripeptides can be prepared out of 3, that is 3 different amino acids. If it is given, you can go for the 6 sets of tripeptides you can prepare. That is the answer. If it is 2 is given, you can prepare for the AB and BA. AB and BA, these two are different. If 2 amino acids are given, you can have 2 different dipeptides. If 3 is given, 6 comes, like that and so on. Next. A codon has a sequence of A and specifies particular B that is to be incorporated into C. So, a codon has a sequence of amino acids. A is a amino acids are bases. Codon will have a bases. So, a codon has a basis and specifies particular amino acids that is to be incorporated into a protein. So, A is a basis, B is a amino acids, C is a protein. So, therefore, it goes to the fourth answer, 3 bases, amino acids and protein. So, that the fourth answer is the right one. Next. Parkinson disease is linked to abnormalities in the levels of dopamine in the body. The structure of dopamine, it is actually it is a Parkinson means it is a nervous system breakdown, it is and body hands will be going on shaking, and this is a that is L-dopa is the medicine used, that is also called dopamine here. Its structure is a benzene with OH, 2 OHs and the propyl amine, that is structure 2 is the right answer. Next. Match the following column. So, oxyhemoglobin I think right answer is missing here. Actually, oxyhemoglobin will have a Fe plus 2 paramagnetic. A goes with 5. A goes with a 5. Aspirin is analgesic. B goes with 1. And hemoglobin is a oxygen carrier. Hemoglobin is a oxygen. C goes with 2. And chlorophyll is photosynthesis. In the preparation of the photosynthesis, we will get a carbohydrates preparation. Next. Hydrolysis of sucrose with dilute aqueous sulfuric acid. Sucrose is a disaccharide which contains 2 monosaccharides, glucose and the fructose. So, therefore, glucose fructose is the first answer is the right one. D glucose, D fructose. Next. Which of the following biomolecule acts as a specific catalyst in biological reactions? The proteins actually, the proteins in having the, that's what I said, primary, secondary, tertiary, quaternary acts as a biological catalyst is known as enzymes. Enzymes are made up of with the essentially proteins only. So, the fourth answer is the right answer. Next. Which one of the following statement is not correct? In oxyhemoglobin, Fe plus 2 is paramagnetic. During respiration, the size of Fe increases when it is changes. That's the right statement. 4 heme groups are present hemoglobin. That's also right. Heme is the prosthetic group. It is in the non-protex part. So, second and third and fourth are right except the first one answer. Okay. Iron is a diamagnetic actually. Next. Natalite is used in the, it is a substitute for petrol actually. So, it is a substance for the used substitute for the petrol. It is in the hydrocarbon combustion type. It is working. Next. Which of the following is not correct? Chlorophyll is responsible for the synthesis of carbohydrates is right. The compound formed in the addition of oxygen to hemoglobin called oxyhemoglobin right. Acetyl salicylic acid is known as aspirin right. The metal ion present in vitamin B12 is magnesium 2 is wrong. In chlorophyll magnesium 2 is right. Vitamin B12 contains a cobalt plus 3. So, the answer 4 is wrong. Next. Fluorosis disease is caused due to the reaction of the calcium with excess of fluorine in the body. Reaction of calcium deficiency leads to this the bones will go on the twisting and all that. So, the answer 1 is the right answer. Next. The reagent used in the preparation of aspirin from salicylic acid, that is we are acetylating actually. Aspirin is nothing but acetylated salicylic acid. Take salicylic acid and add acetic anhydride. Answer 3 is the right answer. Next. The metal ion present in vitamin B12 is cobalt plus 3, that is 1 is the right answer. Next. The name also cyanocobalamine, cobalt is there. Which of the following is used as a rocket fuel? The hydrogen gas with oxygen is used as a rocket fuel. Next. Propellant, it is also called propellant. Next. The blueprints are based on the formation of transition metal cyanide complex. The metal is a chromium, I think. Iron, oh I see. Iron gives a blueprints. Next. Which of the following is a monosaccharide? Means on further hydrolysis you should not break out. Sucrose is a disaccharide, lactose is a disaccharide, contains glucose and the galactose. Maltose is 2 glucose units. Fructose is a single unit, that's why it is a monosaccharide. Next. Following is not example for polysaccharide. Lactose is disaccharide, starch is polysaccharide, cellulose is polysaccharide, glycogen is also a polysaccharide. Lactose contains glucose and the galactose is a disaccharide. So, therefore, answer 4 is the following is not example for the polysaccharide. 1 is the right answer. Answer is given wrong. Lactose is the right answer. Lactose is the right answer. Next. A tripeptide contains a 2 number of NHCO peptide bonds, 2 number, that is second answer is the right. Next. 2 peptides, but 3 amino acids will be there. Okay. 3 amino acids, two peptide bonds will be there. Next, sequence of amino acids in protein is known as a primary structure. Primary structure is the right answer. First one is the right answer. Next, vitamin C deficiency leads to scurvy, that is teeth will have some problem, that is known as scurvy disease. Next, ascorbic acid is the vitamin C is known as. Cyanocobalamin is also known as vitamin B12. B1 is a thiamine, B2 is riboflavin, B6 is pyridoxine. Each one will have its own the names and the biological action. You can see the table. Next, match the following. Ascorbic acid is vitamin C, retinol is vitamin A, calciferol is vitamin D, riboflavin is a B2. So, the answer goes to the second combination 3, 1, 4, 2. Next, a biomolecule with a sugar unit and a heterocycle base is known as a sugar and a heterocycle unit is called nucleoside, that is called. Answer 3 is the right answer. If phosphate is there, it is called nucleotide. If it is polymerized, you will get a nucleic acid, right? Now, next, which of the following is not a pyrimidine, okay, PY, pyrimidine. So, pyrimidine is a, adenine is a purine, so rest are the pyrimidines. Next, deoxyribose is present in DNA, that is what D stands for, the deoxyribonucleic acid. RNA stands for the ribonucleic acid, DNA stands for the deoxyribonucleic acid. However, the adenine triphosphate, adenine diphosphate, ADP are just like the is only bases with the phosphates, that is all. Second is the right answer. Next, faster. The order you find in nucleic acids is sugar, phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, or sugar with phosphate and base. Actually, a sugar is linked to the phosphate, and sugar will have a base hanging. Actually, that is the right answer. Slightly the order is written in a different way, but sugar, phosphate will be there. Sugar will have a base hanging group actually. Right. Next, the three-dimensional structure of DNA described as a, the three-dimensional structure means double helix it is. Two DNA strings will be go on twisted on the opposite direction. Double helix structure it is. Next, the simple lipids are triglycerides and waxes. The compound lipids are phospholipids and glycolipids. The derived lipids are includes steroids and prostaglandins are example for the they are called the derived lipids, in fact. Fourth type, derived lipids. Next, the biomolecules which are produced by ductless, DUCT, ductless glands are hormones. That is answer 3. Next, insulin that maintains glucose level in blood is secreted by the gland called pancreas. Pancreas produces the insulin, is a proteinous material, it is a hormone. Next, around 13 proteins will be there. Which of the following is a tranquilizer? Tranquilizers are they act on the central nervous system, releases some tension and you will get a relaxation. They are badly affecting on the central nervous system, but temporarily you will get a relaxation. Barbituric acid and luminol and Valium, Seconal are the tranquilizers. Ibuprofen is the analgesic drug. So, second answer is the not tranquilizer it is. Next, 38. Cyanochloroxylenol and bithionol come under the this is antiseptics. They are antiseptics. Antacids are omeprazole. Artificial sweeteners, you know, saccharin and all that. Next, Which of the following is not an artificial sweetener? Aspartame, alletane, sucralose are these answers for them. But the fourth one, omeprazole, is a anti-acid it is actually. So, answer 4 is the right answer. Next. Penicillin, chloramphenicol and sulfadiazine are the antibiotics actually, antibiotics. This is not correct actually, antibiotic, answer is antibiotic, which is not there here, wrongly printed. Next, that was the about the all different molecules which are present in the chemical molecule, that is a molecule, especially organic molecules which are present in the body and also used in the different forms of drugs, medicines and all that we have seen today. In the last next tomorrow we will have a class on the qualitative and quantitative analysis. That will be the last class for the organic chemistry that we will take up tomorrow. Thank you.